Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. I hope you guys understand these videos are intended to provide a foundational understanding of some of the basic principles in microbiology. By no means are they intended to be the full detail of all the gory information that we could go over. There's far more information than we can go over in a single semester. You can be studying these things for 20 years and still not understand all the information. So we're going to provide the foundation so that you guys can add details as you go on in your education, and they will make sense. Now, the last video we started talking about the genome, and we did a bunch of definitions on genome and genetics and heredity, and we talked a little bit about prokaryotes versus eukaryotes. Prokaryotes have many linear chromosomes, multiple alleles because you get two forms of every chromosome. For prokaryotes, there's a single circular chromosome, so you get one allele, one shot at every gene. We said that a gene was a series of bases on a chromosome or a series of nucleotides on a chromosome. That's the instructions for a single protein. And prokaryotes, bacteria, can carry extra chromosomal pieces of DNA called plasmids. Now, I want to get into the structure of DNA today so that we can understand how all of this stuff works. So this works for both prokaryotes and eukaryotes. It turns out that all life forms that we know of in the universe utilize the same chemicals, the same molecules for heredity, for DNA and RNA, for proteins. The amino acids used in our bodies are the same 20 amino acids used by plants, by coyotes, by roadrunners, by Bugs Bunny, it's true. By prokaryotes like E. coli and Bacillus subtilis and Staphylococcus epidermis and Staphylococcus aureus. All living organisms use the same genetic code, the same basis for DNA, RNA, and the same amino acids for proteins. So the genetic code is universal amongst living organisms, as far as we know. Now, when it comes to the structure of DNA, one of the things that we know is that DNA and RNA, which in general we'll refer to the two together as nucleic acids. They get their name because they have an acidic nature to them. They have they're acids. Can't speak right now. And they're found in the nucleus of organisms prior to the discovery of prokaryotes. And so the term has stuck. Okay, as you know, prokaryotes don't have a true nucleus, they have a nucleoid region. They don't have any membrane-bound organelles. Now, both DNA and RNA, or the nucleic acids, are also thought of as to be as polymers of these things called nucleotides. Okay? Well, all the nucleotides that we're going to talk about consist of, or are made of, the same three things. One of those things is called a 5-carbon sugar. Now, when we talked about glucose, when we talk about galactose, when we talk about fructose and sugars that are used for energy production, those tend to be 6-carbon sugars. But for the structure of DNA, we use sugars that only have 5 carbons in them. There's still some oxygens and hydrogens. All sugars have carbon, oxygen, and hydrogen, but these are only made up of 5 carbons. A second structure or molecule that's found in DNA and RNA is called phosphate. And we're going to talk about phosphate in a minute. And the last thing that is found in there is what we call a nitrogenous base. Okay. A nitrogenous base is something that has a basic pH and contains nitrogen. It's got carbon, hydrogen, oxygen, and nitrogen in it. And despite the fact that there's a lot of bases in DNA and RNA, there's a lot of phosphates or phosphoric acids that will cause the nucleic acids to have more of an acidic nature to them. Okay. Now, when it comes to DNA and RNA, if I were to put this in a nice, neat little table, we can basically make some simple distinguishing we can distinguish between the types of components that make up each nucleotide for DNA and those from RNA. In DNA, the 5-carbon sugar is called deoxyribose. You should learn after a while that anytime you see OSE at the end of a word in chemistry or biochemistry, we're usually talking about a sugar. Glucose, fructose, galactose, lactose, anything ose, mannose. The 5-carbon sugar found in RNA is called ribose. And the difference between these two is that this one is missing an oxygen molecule. It's one oxygen away from being ribose. But it makes a significant difference in the structure of DNA and RNA. Now, the phosphate group for DNA is usually a phosphate with 4 oxygens and a negative charge. And it's bound to the rest, some other piece of carbon over here. And it's the same phosphate, or PO4, that we use for ATP. It's the same between DNA and RNA. Phosphate is phosphate is phosphate. The nitrogenous bases, there's 4 of them in DNA. One is called adenine, which is abbreviated with an A. One is called thymine, which is abbreviated with a T. Another one is called guanine, which is abbreviated with a G. And another one is called cytosine, which is abbreviated as a C. So we'll see these bases abbreviated as A, T, G, and C. So if I wanted to build a nucleotide for DNA, I need some deoxyribose, I need a phosphate group, and I need one of these 4 nitrogenous bases. Now when it comes to RNA, and I didn't draw this very well, but for RNA, it has the same bases. It has adenine, guanine, and cytosine. And I'm not going to write them out, but those are the same 4. But one thing that's unique about RNA is that there is no thymine present. It uses a thing called uracil, which we abbreviate with a U, in place of thymine. So DNA has 4 nitrogenous bases that can be used: adenine, guanine, thymine, and cytosine. RNA uses 4 bases: adenine, guanine, and cytosine, identical to DNA. But in RNA, there is no thymine. There's only uracil, okay, in its place. So if I were to try to build a polymer of DNA, I could do it this way, okay? So I'm going to erase all this, and we're going to discuss the structure of DNA. So when I say a polymer of nucleotides, first of all, let me take a bunch of individual nucleotides. If I want to build a nucleotide, I need 3 things. And one of those things is going to be a 5-carbon sugar. Another thing is going to be a phosphate group. And then sticking off of that, let me draw it this way. Sticking off of this, so the way that the carbons and the hydrogens and oxygens on the sugar are arranged is such that this sugar can bind to a phosphate group. I'm not going to go into the chemical arrangement. You can look that up very easily. And then I'm going to take the base adenine. So now I have a nucleotide, a sugar, a phosphate, and a nitrogenous base. Now, do we know if this is an adenine for DNA or RNA? Well, it depends. I haven't drawn the structure here, so we don't know. We could look at it, but I want to show you something in a second. If I took another sugar, another phosphate, and another base, cytosine, sugar and phosphate and guanine, and sugar and a phosphate and another guanine, and another sugar and another phosphate and another adenine, and then let's do one more, a sugar, a phosphate, and a thymine. Well, I certainly know that if there's thymine present, since thymine is only available in DNA, that sugar has to be deoxyribose. For the others, I'm not sure if it's deoxyribose or ribose. Now here's the deal. Phosphate can form a chemical bond with more than one sugar, either deoxyribose or ribose. And those 2 sugars can form a bond with more than one phosphate. And if I were to go in here and create a chemical bond between all of these, I have now strung them all together. Now I have a strand several nucleotides long. I have a polynucleotide. And I happen to know this one must be DNA because I see thymine in it. If thymine is present, then all of these sugars must be deoxyribose. So now, one of the things we know about DNA is that DNA is usually double-stranded. And we're not getting into the organic chemistry and the biochemistry, but those are important courses that you'll take eventually, hopefully, if you move on in your education. Now, if you do the health sciences, you're not going to need to know all of this in such detail, so you don't need to take the organic and biochem. But these molecules have a three-dimensional shape to them and orientation. And it turns out that adenine can form chemical bonds with one of the other four bases. And we're going to talk about the base pairing rule. Because since DNA is double-stranded, I'm going to have a series of other bases on the other side. So what I want to do is this. I want to draw the complementary strand. I have a strand of DNA here. It's a single strand. DNA is usually double-stranded. And when I see complementary with an E there, that means what goes along with it. Complement with an I means, wow, you really have pretty hair, or that's a nice car. With an E, those are two things that complement or go well together, like peanut butter and jelly, or peas and carrots, or Steelers and Super Bowl wins. So, for all you non-Steeler fans out there. Anyway, if I wanted to draw a complementary strand, there was a guy named Erwin Chargaff that figured out what they call Chargaff's rules of base pairing, or we're just simply going to call it the base pairing rule. And the base pairing rule is based on the three-dimensional arrangement of adenine, thymine, guanine, and cytosine, and how they can form hydrogen bonds with other molecules. And it turns out that adenine's structure is such that it can form two chemical bonds with thymine, and only thymine. Adenine will not bind to another adenine. And part of the reason is, it's kind of like trying to put my index fingers and my pinkies together to form a chemical bond. Now, the way that adenine is structured, another adenine might fit like this, and it wouldn't work. But thymine is oriented in a way that they actually fit together. So adenine does not bind to adenine. Adenine will only bind to thymine. It will not bind to guanine. It will not bind to cytosine. So there's a rule in chemistry that says adenine will bind to thymine and vice versa. Well, it turns out that guanine has the ability to form three chemical bonds with only cytosine, and vice versa. So everywhere that I have an adenine on one strand of DNA, the complementary strand must have a thymine in its place. Everywhere I have a thymine, the complementary strand must have an adenine. Everywhere I have a guanine, the complementary strand must have a cytosine, and vice versa. So if I know this base sequence, then I could go in here and I could draw my complementary strand. I automatically know the order of bases on the complementary strand. And then I simply know that this is bound to a sugar, which is bound to a phosphate, which is bound to a sugar, which is bound to a phosphate. And we call this the sugar phosphate backbone of DNA and RNA. And what I've essentially created now is a double-stranded DNA molecule. And because scientists know once they see the thymines that this must be deoxyribose, and they already know that this is a given, it does not change from one strand of DNA to another, when we actually sort of use a shorthand to write this, someone might write this sequence like this, A, C, G, G, A, T, and the complementary strand would be this. We don't even put the base pairs in there sometimes, and sometimes they do it as a single bond, because it's understood that there's two bonds between adenines, three bonds between G and C, but scientists don't always want to do all that notation once we understand some fundamental principles. So now, DNA can look like sort of a ladder type structure. Well, it turns out that molecules of DNA with all the base pairs that we have in the human genome, by the way, humans have 3.2 times 10 to the 9th base pairs. So there's actually 3.2 billion of these bases if you add up all the chromosomes together. And because of the spacing and the size, if we strung it together, it would be over 1 meter long. And as you know from looking at a microscope slide, even when we magnify cells 1,000 times, they're not much bigger than my pinky nail. So how do we fit all of these bases in there? Well, one way is to coil the DNA up. And so if I grab the two ends of this DNA molecule and I twist them in opposite directions, what happens is the DNA gets twisted and it looks like a twisted ladder sequence. We have the sugar phosphate backbone on the outside, and then on the inside, we would have our A's and T's and G's and C's with their little bonds in here. And so it's literally like a twisted ladder. It has sort of a three-dimensional shape to it. It's really thick, for example, here, and then we'll do this. And so it's kind of hard to demonstrate on a flat board, but there is a three-dimensionality to this, and all the bases are in the middle. And if I continue to twist this and twist this and twist this, it will compress and compress and compress. In addition to that, once it's all compressed, we wrap it around special proteins called histones, and we can really compact the DNA. So, DNA exists in the form that we call a double helix. The double helix model of DNA simply states that there are two strands of DNA that are coiled around each other. They're helical, and there's two. And so they're literally wrapped around each other like this. The bases are in the middle, and the sugar phosphate backbone is on the outside. Now, understanding this information was a revelation to scientists back in the 1950s. And what we started to understand is how could DNA be replicated so that when a cell is reproducing, that we can copy the DNA and put it into one copy into the new cell, and the old cell remains with its original copy. Excuse me, after doing so many lectures in a row, my voice gets a little hoarse. Now, in RNA is usually, and I hate when books say this, but they say it's usually single-stranded, and it can be. But RNA can be single-stranded or double-stranded. And we'll talk about how and why in a little bit. Now, one of the things that this allows us to understand when we understand the structure of DNA is that DNA can be replicated, which simply means copied. We're going to talk about DNA replication, and DNA replication is said to have a semi-conservative replication, or there's a semi-conservative nature to replication. So what this means is it is partially not changing, okay? And here's how and why. So I'm going to take a double-stranded DNA molecule, and it's going to be this long coil of bases like this. And remember, these chromosomes are really, really long. Thousands of base pairs for a single chromosome. And this happens in both prokaryotes and eukaryotes. The process is slightly different, but nonetheless. When a cell gets ready to divide, somehow, some way, some enzymes can bind to this. And there's several enzymes that bind to the DNA. And once they bind the DNA, they're going to uncoil it and then break the hydrogen bonds and start to unzip it. And we don't do the entire molecule all at once. But let's say we started at this end. What I would do in DNA replication is we were going to uncoil and unzip the molecule. And that's really kind of how we refer to it. And so our double-stranded molecule will end up looking like this. It's coiled around each other, but then there's a part that is started to open up, and it almost looks like a two-pronged fork. And they call this the replication fork. And at the replication fork, what we will see is several enzymes involved here. Now, one of the enzymes that binds to DNA is going to bind here, and it'll be moving in this direction, And this enzyme is one that uncoils the DNA. And so we call this DNA gyrase, or sometimes we just refer to it as gyrase. And gyrase will uncoil the DNA. That's its job. Behind that, we have some bonds. And let's say I have some A's and T's and C's and G's here, with all their chemical bonds. Well, if I have more of them here, oops, I didn't do my complementary strand right, did I? Well, there's another enzyme that's in here that zips along, and this enzyme is called helicase. And helicase unzips the strands. Essentially what it's doing is it breaks hydrogen bonds between the two strands. These little bonds are hydrogen bonds. Now earlier in the semester when we did our chemistry stuff, we talked about the different types of chemical bonds. Hydrogen bonds are very weak bonds. And so DNA gyrase will start to uncoil the DNA, and it starts to zip down the molecule, and behind it, this is a complex of enzymes. Helicase is coming in, unzipping and breaking these chemical bonds. Now, one of the things that I haven't mentioned and I'm going to mention is that DNA has what we call a 5' to 3' nature. So I'm going to explain this in a second. Let me follow my strand for a second. So this one's going to start at the 5' end, this is going to start at the 3' end, and as I go down my strand, this is going to be the 3' end of this strand, this will be the 5' end of this strand. What do I mean by that? Well, when we look at the sugars that are making this up have 5 carbons. I'm not going to draw all the bonds and all the hydrogens, but everywhere that there's a point here, there's a carbon. I have an oxygen here, and then I have another carbon group here. And then this carbon group is going to have an OH on it. And again, I'm not drawing the complete structure, I'm just giving you an idea. And because this is going to bind somewhere else, the way that we name them is this is the 1', the 2', the 3', the 4', and the 5' carbon. And it turns out that this 3' carbon is going to go bond with the 5' OH. And when they do that, this one will lose an oxygen and a hydrogen, and this one will lose a hydrogen, and it's going to bind to the oxygen that's on the 5' carbon, that's on the next sugar. Again, 1, 2, 3, 4, and 5. And then this one is going to bind to the oxygens that's on the 5' carbon of the next sugar. Okay? And there's a phosphate in here. I didn't draw this exactly correctly, but essentially we go from 5' carbon to the 3' of this carbon, and then this one's going to bind to the 5' of the next sugar, and then the 3' carbon of this sugar will bind to the 5' of the next sugar, and then over here we'll have this. And if I look at it, on this end, the 5' carbon is free of any other bonds, and on this end, the 3' carbon is free of any other bonds. Now, you don't need to know those details, but the idea of 5' to 3' is somewhat important to us. This is some of the why. Again, I didn't draw the phosphates in here correctly. I made a mistake, but you get the idea. I have a free 5' carbon at one end, and then the 3' and the 5' and the 3' and the 5' are all bound up. But when I get to the other end of the DNA molecule, I have the 3' carbon OH is free on this one. And the other strand is going to run in the exact opposite direction. It's going to run 5' to 3', just when we look at their structure. That's not the best explanation, but just bear with me. So when I talk about 5' to 3', I'm just talking about the orientation of the sugar phosphate backbone. And the reason I mention that is because there's an enzyme that's going behind all of this called DNA polymerase. Now, very often you need to know the functions of these enzymes. Gyrase uncoils or ungyrates DNA and uncoils it. Helicase unzips it. And then DNA polymerase is coming in here, and everywhere it goes, it's going to add in the complementary base. And essentially what it's doing is it's reading a strand, and when it gets to a particular base, let me do this as an example. Every time I have a blue marker here on one strand, that means that the other strand, that's going to represent adenine. And let's say my green marker represents guanine, and my red marker is going to represent cytosine, and my black marker will represent thymine, okay? If I'm reading this strand here, and I'm DNA polymerase, as I go along, let me set these over here, get them out of the way. If adenine is blue and thymine is going to be red, then everywhere I have a blue marker on this strand, I'm going to come in. If I grab the wrong base, let's say I grab guanine for green, and I try to stick it on this adenine, it won't form a chemical bond and it'll fall off. But if I grab the thymine, then when I stick it on here, they'll actually stick next to each other. And for guanine, I can go in and I can take a cytosine and stick it next to it. And vice versa, okay? So I can go along here and actually build my second DNA strand as long as I know the order of bases on the first strand. DNA polymerase will bind another base and try to add it in. If it's the correct base, it will stick. So everywhere that there's a thymine, DNA polymerase is going to add an adenine. And everywhere there's an adenine, it'll add a thymine and guanine and cytosine. And then it sticks these bases, the backbone, together, forming chemical bonds. Now watch. On the opposite strand, I'm going to be I'm just going to put DNA pol for DNA polymerase. I'm going to be sticking the opposite bases here and string them together. So I'm replicating both strands at the exact same time. Now I know you probably can't read those bases very well, but just follow along. Now, over time, what's going to happen is, as I go further down the DNA molecules, I'm going to see that these enzymes have shifted all the way down here. And so my DNA gyrase is still working down the molecule like this. It's still uncoiling the DNA. I still have my enzyme helicase. It's still unzipping bases. And let's say I have a bunch of bases here. And at this point, I don't know, I have C, C, G, and this one's still stuck together, but the others are being broken. Nope. Okay, I'm just putting some random bases in here to illustrate something to you. And I know this is kind of slow and tedious, but if I follow this, on this strand, my DNA polymerase has started to add more bases here. And it's stringing them together, but now it's sticking them to the bases that were previously added. And I don't know, let's say, I don't have the correct base sequence here. I wish I had done it exactly as this, but suffice it to say that this strand is being copied in one direction from 3' to 5' on one strand. The other strand is being copied from its 5' end, from its 3' end to its 5' end, because that's the direction that these enzymes can read. Now here's one of the problems. On this strand, the original DNA polymerase, when it gets down here, may have only built a series of bases here, for a short segment, because I'm unwinding this, but I've already started moving in this direction. And as I unwind further, I gotta have another DNA polymerase here start adding these bases. And so this one strand gets built in short fragments. They happen to be called Okazaki Fragments. So if you hear about this, they're named for the Japanese scientists that figured them out. Okazaki fragments are the short fragments of DNA that are being replicated on the 3' to 5' strand, or in the opposite direction. There's another enzyme, and that enzyme happens to be called DNA ligase. And DNA ligase can ligate, stick together these strands. And so it'll go in and fix any of these broken bonds between the second strand. And eventually this will continue. These enzymes, gyrase, helicase, and polymerase, are all going to work down to this end. And in the opposite direction, I'm going to have another DNA polymerase making Okazaki fragments and ligase sticking them together. And I'm going to erase these two terms because ultimately, I need some room here and I'm running out of room, but ultimately by the time I'm done, by the time these enzymes get to the end of the molecule, and then this one zips down the other end, connecting all the Okazaki fragments, what I end up having is this. Now I'm going to use this base sequence here, but whatever my base sequence was on this other molecule, I'm just going to start from here and copy it down. If it was CCG, TTA, CAT, AAG, CCC, GAT. I'm just going to add some more bases to the end here. And my other original strand then obviously had this, GGC, AAT. I'm simply using the base pairing rule. Those are my two original strands. My new strands will look like this. On this molecule, it will have GGC, AAT, GTA, TTC, GGG, CTA. And if I look at it, this new strand here in green is the exact same base sequence, if I did this correctly, as my original strand or original complementary strand. And so I have, when I'm done copying or replicating DNA, I have one original and one new strand. Hence it's semi-conservative. I've conserved one original strand, but the other strand is new. And on this original strand, when this DNA polymerases are all done and the Okazaki fragments are all linked, I would have a long strand of DNA that would read this. And you get the idea. And it turns out that this base sequence is identical to this other original strand. So I truly have two identical copies of the original strand of DNA. And there's other enzymes that will come in and start to coil it back up and pack the DNA. So in DNA replication, the big picture is this. I'm going to copy a double-stranded DNA molecule, my double helix, using a series of enzymes. The most important ones that I think you should know at this point, your instructor may include others, because there's other forms of DNA polymerases, 3 and 2 and 1. But we have DNA gyrase, which uncoils the DNA, DNA helicase, which is unzipping or breaking the hydrogen bonds, and DNA polymerase is coming in behind, adding in the complementary bases, building my new strand of DNA. On the opposite strand, DNA polymerase is going to be adding in bases in short sequence, and then another enzyme called DNA ligase will connect those Okazaki fragments so that I end up with two identical daughter chromosomes. And each daughter chromosome has one old strand from the original piece that I copied and one new strand of DNA, hence the semi-conservative nature. Now it could be more complex than that, and there's a lot more detail, but I'm not going to worry about it at this time. Okay? So ultimately what you need to know about DNA replication is it's the process of copying DNA into a new strand of DNA. We start with an original strand that's in the form of a double helix. We have some enzymes like gyrase, which uncoils it, helicase, which unzips the hydrogen bonds, and DNA polymerases, which add in the complementary base pairing rule. Based on the structure of the bases themselves, the base pairing rule states that everywhere that I have an adenine on one base and on one strand, I must have a thymine as the opposite or complementary base on the complementary strand. So I can always figure out, if I know one strand of DNA, I can always figure out what the series of bases are on the new strand, okay? Ultimately, I end up with two identical copies, and each new copy has an old strand and a new strand, hence the semi-conservative nature. I think I hit all the high points that I think you should know at this point of your education, at this point of understanding all of this stuff. It's far more complex and involved than this. There are some differences between prokaryotes and eukaryotes, but we're not going to go into all of that just yet. Now, one last detail I'll talk about. I'm going to erase all of this. When we're talking about eukaryotes have multiple strands of linear DNA, just like this. In a prokaryote, you have a single strand of DNA, and then you have the complementary strand of DNA in here. And when we start to replicate, literally the two strands will start to bubble out, and it'll go all the way around. And over time, we add our new strands of DNA, and as we continue to do this, you'll see the two new strands of DNA being added, but eventually, we will get all the way around the circle, and when we do, we peel one double-stranded DNA molecule off the other, and eventually we end up with two double-stranded chromosomes. One will go into one bacterium through binary fission as the other chromosome is moved into the new bacterium. We don't have pairs of chromosomes like we do in eukaryotes. So we still replicate the circle, it's just slightly different. And again, those differences we're not going to get into in great detail. All right, I hope that some of this was informative in your understanding of DNA structure and DNA replication. What is a nucleotide? What are the components of nucleotides? And what are the major enzymes involved in some of the basic steps of DNA replication? So I hope you learned something. I hope you had as much fun as I did. And I hope to see you in the next video. If you like this and you found it helpful, please give me a thumbs up. Hit like, subscribe if you want to see my new videos, and I'll see you in the next one. Thanks for watching.